Hey guys, welcome back to my channel Tuli's Biology. So today again I have come up with a new topic, new idea, that is Avery, Mccarty and Macleod experiment. See, I have done the Griffiths experiment with you people in my previous video. So this is with the Avery, Mccarty and Macleod, and if you haven't seen the Griffiths experiment, I have put down the link of the Griffiths experiment in my description box. So please go and watch the video. And please to all my viewers, I'm telling that please subscribe to my channel, watch the videos, like the videos, share it with your friends. And also if you are having any ideas which will be like beneficial to me, so please share your thoughts. And if you are having any problem, then also feel free to share it with me. So where will you share your thoughts or your problems? In the comment section, of course, or in my Facebook page and mail ID. Do follow up all my videos and keep supporting the channel Tuli's Biology. So let's just start with our topic, that is Mccarty, Avery and Macleod experiment. So in the Griffiths experiment, we have seen that he has performed that experiment, and that experiment was like he told that R2, which is non-virulent, has changed to S3 virulent type. So he said that there was the mixture where he put R2 and it killed S3, and suddenly at the end of the experiment he observed that R2 has changed to S3, not hit kill but directly to S3 bacteria. But how is it happening? So he concluded that yes, R2 has absorbed something from this S3 and changed itself into S3 virulent type. So he could not explain that what they actually absorbed. So this was being explained by Avery, Mccarty and Macleod in the year 1944. So they performed this experiment not within the mice, they performed this experiment in vitro. In vitro experiment means outside the body in the cultural medium. Okay, so they have taken this S3 R2 virulent type. They just done the mixture, and what they see is R2 and hit killed S3. They have done the mixture and they have taken it in a test tube, and they just extracted out all the materials from the from this bacteria. Okay, so from that extracted portion they remove out carbohydrate and lipids. So only what things were there in that extract: protein, RNA and DNA. Only these three things were there, rest of the things were being removed out. Okay, because the scientists at that point of time were not sure that which one actually is the genetic material. Is it RNA or is it protein or is it DNA? Okay, so there was a confusion. So in order to clear up the confusion, they only kept this protein, RNA and DNA, and all the other things they just removed out. So they have taken this extraction in 3 different test tubes. Okay. Now first test tube, they have put one enzyme termed as protease. In the second test tube, they have put one enzyme called RNase. And in the third test tube, they have put one enzyme called DNase. Now protease is the enzyme which will digest the protein. Okay. RNase is the enzyme which will digest the RNA, and DNase is the enzyme which will be digesting the DNA. So after this protease, they saw, or they just transform it in the cultural medium. So after the action of the protease, they transfer this material or this extraction in another cultural medium, and they have seen yes, the transformation has taken place. So yes, transformation occurs. Okay, that means protein is not the genetic material, because if it would have been the genetic material, there is no protein. So the transformation should stop right over there, but it is not stopping, it is occurring. So that means it is clearly proved that no, protein is not the genetic material. Second, they just performed this same experiment with RNAs, that is the RNA digesting enzyme, and similarly they have seen again that the transformation is again happening. That means it is again evident that yes, RNA is also not the genetic material, because if it would have been the genetic material, then the transformation wouldn't have been done, because RNA is not there. RNA is totally digested by RNAs. Next, they performed the experiment with DNAs, that is DNA digesting enzyme. So here they have seen that no transformation takes place. So it's clear that as no transformation takes place after the digestion of DNA, as DNA is absent, no transformation has taken place. So it is evident that yes, DNA is the genetic material. So they just concluded that yes, DNA is the genetic material, whereas protein and RNA are not. They are not genetic material. Okay, so in this way they just proved the fact that yes, DNA is the genetic material. Later on there are other experiments which is done by Hershey and Chase. It is popularly termed as Hershey and Chase experiment. They also confirmed it with very strong points that yes, DNA is the genetic material, none other than DNA, nothing is the genetic material. So in my next video I will come up with this Hershey and Chase experiment. So till then everyone, stay tuned to my channel Tulis Biology and take care of yourself. Bye bye everyone.